Good morning, everybody. Here we go. I have no disclosures. This is the brief outline of my talk today. I'll discuss a little bit the challenges of imaging the developing brain, then introduce Infant FreeSurfer and some of the concepts using surface-based analysis and surfaces in case of infants, and then show you two different applications, so group analysis and then individual analysis using the derived surfaces. So first I wanted to show you this picture. If you are not used to looking at infant brains that are developing, they represent a significant challenge when we are trying to write post-processing tools. And I think this figure illustrates some of the challenges quite well. So here you see images taken of a single subject at birth at 1 year and 2 years of age. In the top row, you see T1 weighted images, in the middle, T2 weighted images, and then a basic 3-4 label segmentation in the bottom row. And so you can see that there's a huge size difference, even during the development of 2 years. So there's a tremendous growth postnatally during this time. So your tool has to be robust to such size differences. You can also see that there's a contrast flip that happens somewhere between the 5 and 7 month, depending on which modality you are looking at. So compared to the adult contrast that you might be more used to, which is probably in the last column. If you look at a newborn brain, that contrast is flipped. So whatever is light, for example, in the T1 weighted image, the white matter in case of adults or the 2-year-old is indicated a darker color in case of a newborn. And the other way around, so the dark colored cortical areas are lighter in case of a newborn. And then also development is still happening. So your tool again, if you're writing an analysis tool, that has to be robust and adaptable to that. A couple more images indicating challenges. So I already talked about contrast, but we can have motion artifacts corrupting the images in case of infants. In case of infants, depending on the age that you are specializing in or recruiting to be imaged, there are certain strategies that you can use in order to not have the infant move. But for most of the studies, we don't use sedation, so the babies can often wake up, so the motion artifacts can be quite considerable, or you have to cancel your scan. Even though we have special baby coils and the brains are padded, or there's certain padding that we use in the coil, there can be still significant changes in pose, especially with the up-down position. So that can pose some challenges. And then also, we often see additional anatomy in the scans. So for example, an infant sucking a thumb, or we can see shoulder and chest included in the brain images. So again, any kind of post-processing that you are doing or pre-processing to your follow-up task need to be adaptive to that. So an intro to FreeSurfer. So you have heard about FreeSurfer before in the previous slides. So the goal when developing Infant FreeSurfer was to develop an image processing pipeline that would be particularly useful for these initial first 2 years. The modality of interest at the time of development was using T1 weighted MR images. And we wanted to keep to the FreeSurfer formatting and processing outputs so that later on, with any kind of longitudinal analysis, we can easily connect the different outputs and do easier comparison in case of multiple time point analysis. I'm going to show you the individual computational blocks and then focus on the surface-related steps here. So this is a quick outline of what FreeSurfer and also Infant FreeSurfer are doing with the main computational blocks. So T1 weighted input image, you do some intensity normalization, some corrections, and then skull stripping, basically getting rid of everything in the background. that's not the focus of your analysis, then there's some volumetric segmentation, basically aligning annotations, anatomical annotations to various brain areas. And then you do surface extraction and then do the parcellation, as you have heard in the talk before. So just a quick terminology: segmentation, parcellation, they are both annotations. In case of FreeSurfer, we usually refer to segmentation as the subcortical segmentation or annotation of areas, parcellation to the cortex, but these should be interchangeable. This is just our particular lingo. You have hopefully by now been convinced that if you're analyzing and interested in measurements that are related to the cortex, you should do your analysis on the surface. I like this image from the Sereno 95 science paper, which shows you basically the functional mapping of primary visual areas, both in the volume and on the surface, on the flattened surface. So while you can see the measurements in both cases, so on the right-hand side, you see three orthogonal views from the volume. It's really hard to appreciate how nicely organized the visual areas are, while if you do the flattened surface representation, that organization is much clearly visible. So in order to be able to keep that organization or being able to visualize what you're looking at in these areas, we also recommend using the surface representation. So running Infant FreeSurfer is fairly straightforward. In the most basic runs, you have two inputs, your subject, and you have to indicate the age at scan information. There's some assumptions about where you store your input images if you're using the default setting. Otherwise, you can specify that. And just like FreeSurfer, there's going to be a very organized output folder structure that's going to be the output. So your subject directory can contain multiple subjects. Your subject ID corresponds to the particular subject you are currently organizing. And then the following 5 folders are going to be created. MRI, which contains the volumetric representations of your analyzed data. SURF, corresponding to the surface-based files. Label, these are going to contain the annotations or the parcellations that go or map onto your surfaces. Some statistics and some logs that are related to basically summaries of all the processing stuff that you have done through the analysis. The 2 main surfaces that we recover are called white and PL surfaces. So on the right-hand side, I showed you their placements. So the white matter surface, as the name indicates, it's the boundary between the white matter and the gray matter. And the PL surface is the one that separates the gray matter from the CSF. I'm lucky because you've seen some of these FreeSurfer pictures before, so you might be already used to the color coding, the default color coding of these images. So on the left-hand side, I'm showing you the PL and white matter surfaces with the FreeSurfer color coding on the top, the white matter surface on the bottom, the PL surface. So how does the surface extraction step happen? As I mentioned, in the Infant FreeSurfer pipeline, you first do a volumetric segmentation. And then for the volumetric segmentation, you assemble a volume called a filled volume. Basically, you're interested in the white matter, and then you're filling up some of the ventricles. And the reason why you do that is because the initialization of your surface reconstruction is going to be aided by this volumetric segmentation. And what we call here, and you see it's the original surface. So basically, this is just a surface that gets fitted to the white matter. And this is going to be, this is not corrected yet. This is really just the initialization of where you want your white matter surface to be placed. And throughout the processing, we are going to correct this, make it topologically correct, and also correct the placement of the surface. We are going to nudge it according to some of the T1 weighted intensities and the volume. There's some smoothing constraints, but throughout the processing, the OH surface and your final resulting white matter surface always have a one-to-one correspondence. This is going to be true for all the various surfaces that I'm going to mention to you. So, and this is very important because if you're working on one surface, you can always propagate the labels or visualize it in another surface. So you are going to see that in some cases, working in the background or with the registration that you just heard about, working in one representation is much easier. But visually, when you are representing your data, it's going to be more useful to use, let's say, the inflated surface or the white matter surface. Given that there's a one-to-one correspondence, it's going to be very easy for you to do that because you can map any of those measurements to any of those surfaces. So we have our white matter surfaces. These are going to be stored in the surface folder. And then this is going to serve, after you find the white matter surface, this is going to serve as an initialization to your PL surface placement. Again, you do the initialization, and basically you push the PL surfaces out from the white matter surface, again using T1 weighted intensity information in order to determine where to stop the placement of the PL surface. And again, we keep the vertex identity, so again, you have one-to-one correspondence between them. So now you have the PL surfaces and the white matter surfaces. And you have seen this mesh model before, but I wanted to emphasize, so our surface model is basically a dense mesh. We call a vertex is a key component, so instead of a voxel, which is an element of the volumetric representation, we work with vertices here, which is a point of triangles. It has a neighborhood that characterizes it, and we characterize the vertex location by x, y, z coordinates. In case of the infant brains, we have about 220,000 triangles that represent the brain, and 110,000 vertices. These have various features associated with them. So for example, area, distance, curvature, and thickness. And I'm going to talk about those a little bit more. You have heard a little bit about volume and surface comparison. So I just have a short list here as well. So in case of a volume here, you have a uniform grid. It basically overlaid over your input image. You definitely have a denser grid here. This is just for illustration purposes. Voxel lies at an intersection of these grid lines. So we usually, in the data coordinates, refer to these voxels by the column row slice indices. So if you want to compare one voxel location to another. The voxels have their sizes. So the voxel has three dimensions. We can compute distances between them, and they have values assigned to them. So either intensity values or vectors of values, depending on what kind of image modality you're working with. And in the 3D coordinate system, we identify them by the XYZ coordinates. On the other hand, with the surface modeling, the grid is non-uniform. The vertex, as I mentioned in the previous slide, lies at an intersection of triangles. Each vertex, in our case, has an index, and these indices, as I mentioned, are going to be one-to-one, have a one-to-one correspondence between the different representations. On average, the distances between these vertices is about 1 millimeter. Each vertex here can be assigned a label or a value, and I'm going to show you what default values they get assigned when you go through the Infant FreeSurfer or FreeSurfer analysis. And again, they are characterized by the XYZ coordinate. So upon completion of the pipeline, I mentioned that your surface directory is going to be filled by various different surface representations. So here we are processing left and right hemisphere separately. So you have the original surface, the white matter surface, PL surface. The inflated surface, if you imagine the little cow that was being inflated, we usually try to talk about kind of a paper bag that gets scrunched up, and then you blow air into it. And so the inflated surface, as you can see, helps you see the details a little bit more. So basically, you don't have information that's hidden on the surface. And most of our algorithms that take place after we reconstruct these surfaces are in the spherical space, so which is basically mapped onto the unisphere. But this is a little bit difficult to kind of visualize findings on, when you're discussing it with neuroscientists or physicians. So it's nice to have an option of going in between the different representations, depending on what you want to do with the surface or what you want to visualize. The parcellations that are basically segmentation labels that are mapped onto these surfaces are going to be located in the label directory. And we have various versions. One of them is the Desikan-Kiliani atlas that you have already heard about in the previous talk. So as I mentioned, with Infant FreeSurfer, you can process a bunch of various ages. So here I have an example of a newborn 12-month-old and an 18-month-old with a PL surface, spherical representation, and the parcellations. And now I'm going to tell you a little bit about the various features that also get computed while you compute the pipeline. So one measure that we compute is the curvature measurement. So at each vertex, we can fit a circle tangent to the surface, and the curvature measurement is going to be the 1/, radius of this circle. There's going to be one single value assigned per each vertex. It's going to be a sign metric, so in order to differentiate between the sulci and gyri. And these feature maps can be found in the LH or RH curve files in your surface directories. Again, this overlay, we call it overlay, so you can map it on your various surfaces, the inflated surface, the white matter surface. This red-green is the default color map that we use in FreeSurfer, but you have a choice to choose that if you wish. We also have area and volume measurements. So each vertex has an area assigned to it, and also a thickness measurements. This thickness measurements computes the distance between the white and the PL surfaces. And the volume is basically the area times these thickness measurements. So when you have these measurements, then you'll be able to do group-based analysis. And I'm going to have kind of a quick demo of how you can organize your data in a way that you can easily do that in FreeSurfer. So again, these features or these overlays can be found in the surface directory as well, just like all the other surfaces. So as I mentioned, you can have these overlays or measures that you have just computed on any of the surfaces. Here I'm showing you on the top row everything mapped onto the inflated surface. So the sulcal measure, the curvature measure, and the thickness measures all indicated in the left hemisphere inflated surface. And the annotations here are on the PL surface on the bottom. As I mentioned, there's a single value for each vertex in case of these feature maps. The color here indicates the value. We have default lookup tables that you can use in order to match previous publications or previous work, but you can definitely define your own lookup table if you have additional labels or you want to distinguish between certain areas. As I mentioned, you can render these on any of the surfaces that you have available. And beyond these measurements, if you have additional measurements, for example, fMRI, various statistical maps, measurements that you derive from diffusion analysis, those can also be mapped onto these surfaces. So Infant Recono was released a while back. We have a wiki page. We have a container-based resources. And we are trying to update that in the background. So, and if you have any questions, please let me know. I'm happy to answer any questions about any of its functionality. There's some ongoing developments that I'm not going to mention too much about, but you have seen that there's a long pipeline with various post-processing steps. So while if you are interested in only using FreeSurfer and get the surfaces or segmentations, you don't need to necessarily know what the background components are. We are trying to improve those to make those more accurate, more efficient, and faster. So we have right now a new skull stripping technology, a new volumetric segmentation, and a surface fitting algorithm. These are all deep learning based solutions. And we are hoping that by midsummer or end of the summer, these are all going to be part of the pipeline. We just need to thoroughly test how each individual step is, has, affecting the final output of the full pipeline. I'm also not going to talk about how we use these surfaces and what kind of questions or group analysis we use. You're going to hear a bunch of talks in the afternoon about how to use surfaces for answering very interesting questions about certain populations. But one of the things that we were interested in doing is creating an unbiased atlas from a neonatal population using cortical surfaces via unsupervised learning. And here I'm showing you, this is another deep learning-based framework, but in the bottom right, I'm showing you age-specific convexity atlas representations at various ages. But I want to emphasize that the tool does not just result in these individual age points. It's a continuous representation. So even if your training data doesn't have representation for certain age groups, it can compute a representation based upon the training data set. So you can represent the full age range of your interest with these representations. Now I'm going to give you kind of a very quick summary of how you can do group analysis when you have used the pipeline in order to generate the surfaces that I mentioned. So the whole framework is going to rely on some of the functionalities that are part of FreeSurfer. But the answer that we are trying to answer here when you are doing group analysis is how does a particular measurement X, for example, change with age? Or how is this different in cohort A or cohort B? You have to identify the surface measurements that you are interested in comparing. So these can be some of the morphometric measurements that we already discussed, or you can have functional PET, MEG, EEG, or some diffusion measurements that you sample under the surface and then project to your surfaces. And then we use a GLM with FreeSurfer, generalized linear model, and I'm going to show you how to define the model and how you can run 3 commands and basically do the analysis. So the nice thing about the structured organization of the output that you get from Infant FreeSurfer and FreeSurfer is that when you process multiple subjects, the corresponding files are all going to be located and named in the same way. So let's say if you're trying to compare thickness measurements between various cohorts or various subjects, they are all going to be located under the same subject directory and at the same location. And in order to, so what you will need to do in order to do this group comparison is to construct a design and contrast matrix. And FreeSurfer is going to help you with that. So we have a file which we call an FSGD file or a FreeSurfer group descriptor file. And FreeSurfer is automatically going to create a design matrix from it. It's a simple text file. I'm going to show you an example of it. It's going to list all your subjects in the study that you want to compare or do the analysis on, and you can include all the accompanying demographics in it. So for example, this is a simple FSGD file. As you can see, there's various classes here. Your class indicates sex, so male or female subjects, and then the various inputs. So here you have 4 subjects, for example, and then indicating the class, which class they belong to, and the various other variables that you include here. I mean, in this study, for example, we have age at scan, weight, and some type of measurement. Note that sometimes you can have very different scales that correspond to your variables. So you either want to normalize those on your own, or you want to include a parameter in this FSGD file that's going to take care of the normalization for you directly. This is a very quick summary of how you need to construct these files, or what information you need to store there, but there's various examples on the wiki, so if you're interested in more complex analysis or details about the analysis, definitely check those out. Now the contrast matrix is going to be basically a vector that's going to summarize the question that you are trying to ask about your cohort. So given that we have linear models here, I have two groups here. You know, each line can be represented by a slope and an intercept. So we are going to translate the question into how they are determined by the slope and intercept differences. So for example, if you want to ask the question whether the groups differ in their intercepts, then your contrast matrix, which is down below, is just going to be a +1, -1, 0, 0. This is going to be your input into the FreeSurfer analysis. Or if your question relates to whether they differ in slope, then there's going to be 0, 0, +1, -1. Again, this is a very quick kind of summary of how you set up, how you translate your questions into contrast matrices. But we have, again, lots of various examples that are detailed on the website. I just wanted to show you how easily you can do that. So then the group analysis has three main commands that you need to run. There's a pre-processing step that basically just gathers all the necessary information from all the subjects and stacks the inputs to be analyzed. Then you run a surf-to-surf command, which is going to do some surface-based smoothing on your data. And then the GLM fit, which is going to produce the eventual T or F ratios and significance maps. And now I'm going to do a quick individual demo, which is going to be demonstrating Freeview. But I had a couple of questions. I think this is not going to work, so maybe I'll try to see whether you can raise your hands. These are not necessarily related to the FreeSurfer talk, but definitely related to development. So I just wanted to wake everybody up before the coffee break. So a quick question: at what stage of fetal development does the brain start to develop? Before conception? Any hands? Okay. 18 days after conception. Okay, you're answering it for everything. And then 5 weeks after conception. So the answer is the last one. It's approximately 5 weeks after conception when it starts. Human brains are large at birth compared to their body size, but I showed you how big of a difference there is between adult brains, or between 2-year-olds and newborns. So what % of the adult brain size do they make up at that point? Guess is for 1/4. Single hand, guess is for 1/3. Couple more, 1/2. Yeah, the answer is 1/4. And when does the human brain reach full adult size? So the answer, 5 years of age. Couple hands, 12 and 20. Okay, yeah. So it's mostly 20. So by age of 5, you reach around 95% of the brain size, but development lasts until much later, so into early 20s. And then this is kind of a throw-off. What is the average age in month of brain samples that are in my lab refrigerator? So as a quick side note, I write algorithms not only for in vivo brain analysis, but also post-mortem. So this is not a sick obsession that I have these brain samples, but we have those for the post-mortem analysis. So this is really just a chance. If you want to guess 1 month, 6 months, 12 months, or 13 years. Okay, so I mostly study infants, so it's 12 months. We are mostly interested in the first year of brain development, but I have a couple older samples, so that's kind of skewed the average. But yes, so these were the questions that were kind of just to kind of check in with everybody's attention. So I'm going to show an individual demo, and the main focus here is that I'm trying to encourage all of you, if you do any kind of analysis, to check your data, before drawing any attention, like any kind of complicated analysis, asking questions about how this cohort compares to the other, or you get very excited about these huge, large data sets that are great that we have access to. Make sure you know what the input is. Make sure that you check, the surface labels, the volumetric labels, or whatever it is that you are going to be working on, because that's very important, right? If you don't check and there's some errors or mistakes, and you don't notice that, then all your analysis is going to be comparing, erroneous measurements to something that you don't know. So I wanted to do a live demo, we didn't get to it. It's going to be, I'm going to talk over a video showing you how you can use Freeview, which is a visualization tool in FreeSurfer that you can independently download, or you can use your own volumetric visualization tool. So this is nothing particular to FreeSurfer that you need to use in order to check your data. We have a bitsified 18-month-old subject uploaded to OpenNeuro, so you can download this data set, if you don't have access to other infant data sets, and you can, play around with it with your, in your visualization tool of interest. Very quickly, before this, I wanted to mention that, as I said, we have containerized versions of Infant FreeSurfer. So there's you can either download or you can construct your own singularity image. We have instructions on the website about that. You need to have a license before running FreeSurfer, Infant FreeSurfer, so download it and store it on your desktop or laptop. And then this is the command that you would use when you run Singularity. Only 6 various parameters to set for the default run, so it should be fairly straightforward. We have more instructions on the wiki again, and if you have any questions, let me know. If you already have FreeSurfer and the infant version downloaded, again, the command is fairly simple, so you just need to set the data location, identify the subject and the age, and then you run the command. The command that I'm going to be demonstrating right now, this seems a little bit verbose here, but you are going to see that on command line, I'm assigning various choices for lookup tables, for colors of the surfaces, whether they are visible or not. Again, if you want to follow this demo with the bitsified download that I indicated, you should be able to just copy and paste this command, and this should be working for you. So let's see whether this is going to start. Can you start? Yeah. All right. So unfortunately, the font size is very small here. The only thing you will be, what I'm trying to demonstrate here is that I ran the command that was on the previous slide, which is it running right now? No. Oh, cannot play media. Yeah. So there's a video. Yeah, there's a video separate. Yeah. Is there a way to speed up, like, I don't know, you can put 1.2 or 1.5 so that we don't run out of time? All right, so, yeah. So here you see the contents of the MRI, the surface directories, and the label directories, just like they were indicated on the slides. So here I'm in the subject directory that was the output from Infant FreeSurfer. And now I'm going to just copy and paste the command that you saw on the command line. So this is a Freeview command. Freeview you can open up by itself. And then if you are used to using the GUI application, just use load volume, load surfaces. In order to make it kind of reproducible and shareable with people, I like to just, have the full command line and, let everybody kind of use the same color maps, the same panel layout, so it's going to be easier to compare the things. So here we read in the normalized, the norm.mgz, which is the skull strip, the intensity normalized volume of your input image. and the volumetric and the full brain segmentation, which is called ASEC for automatic segmentation. So here you have an option of looking at 4 different panels. You have the 3 orthogonal views from your volume, and the third one is going to be a surface 3D representation that we are going to look at in a second. So in the 3 views, you can just kind of zoom through all the different slices. If you did a segmentation, make sure the segmentation labels are located in the right location. If there's a particular area that you are interested in, make sure the boundaries are matching, what you expect to see them based upon the anatomy. And after you are done with that, you can overlay the surfaces. Here I'm changing the opacity for the segmentation so you better see the surfaces. In the 3D volumetric view, you can choose the boundary colors for your PL and white matter surfaces, so it has a better contrast from the background. So now we switch to the 3D volumetric representation. Here we have the PL. So I have highlighted both the PL and white matter surfaces, but given that the PL surfaces are over the white matter, initially you could only see the PL surface representation. Now we unclick those, so you see only the white matter surfaces. And now I made the inflated surfaces to be visible, so you can zoom in on some of the features, you can turn them around, you can look at them one by one. And this is the spherical representation. Again, you're going to see these in publications. They are very useful for background mathematical computations. I don't think this is going to be the space where you're going to be presenting your results, but this is an option. Again, these have one-to-one correspondences, as I mentioned. This is the white matter surface. I'm going to read in a bunch of different overlays. So here in the overlay menu, you can write in, for example, the thickness map. You can, so right now it's displayed over your surface. You can play with the threshold. You can play with the various color maps that you can overlay on the surface. And if you want to compare various subjects, you want to do that step as consistently as possible. So basically the colors are going to be corresponding to the same colors between individuals. The demo here is just overlaying the same thickness map on the various surfaces. So first you saw it on the white matter surface. Now we have it on the PL surface. And then you can do that for the inflated surface. Again, here, this is just another color map that we can use for this feature map representation. So we see the sulk map and here the curvature map overlaid on our inflated surface. It's a slightly different color map that we've used before, but again, you can change that and customize that to your liking. I believe, okay, there's one more. We are doing that on the sphere as well, the curvature measurement on the sphere. Again, depending on what you use these surfaces for, whether you are, this is your end, or you are going to be using these surfaces as input to further analysis. That's going to determine which one you are using. So we can stop the video. I have 2 more slides just to say thanks. So I'm part of the Library of Computational Neuroimaging, and I have, the infant brain imaging group within that. We have various resources about Infant FreeSurfer and FreeSurfer, and please check those out. If you have any questions, let me know. Thank you very much for your attention.